Virus-induced cancer, mechanisms, oncogenes, and viral contributions to tumorigenesis. Cancer development can be driven not only by mutations in host cell genes but also by viral infections. Some viruses have evolved to interfere with the normal functioning of the host cell cycle and promote uncontrolled cell division, leading to virus-induced cancers. These viruses are called oncogenic viruses or oncoviruses, and they can cause cancer by inserting their genetic material into the host cell genome, disrupting the regulation of normal cellular processes. Oncogenic viruses are responsible for a significant proportion of human cancers worldwide. There are two main categories of viruses that are known to cause cancer. DNA viruses. These include human papillomavirus (HPV), hepatitis B virus (HPV), and Epstein-Barr virus (EBV), among others. RNA viruses. These include hepatitis C virus (HCV) and human T-cell lymphotropic virus (HTLV). Mechanisms of virus-induced cancer. The processes through which viruses induce cancer are complex and involve several mechanisms. Viral integration into host genome. Some viruses, particularly DNA viruses, integrate their genetic material into the host genome. This integration can disrupt normal cell cycle regulation, promote the expression of viral oncogenes, or lead to the inactivation of tumor suppressor genes. For example, integration of HPV DNA into the host genome can disrupt normal epithelial cell growth and contribute to the development of cervical cancer. Oncogene expression. Oncogenic viruses may carry viral oncogenes that can directly promote cell proliferation or survival. These viral oncogenes often mimic the function of host proto-oncogenes, leading to the activation of pathways that promote cell division and inhibit cell death. For example, the processes through which viruses induce cancer are complex and involve several mechanisms. Viral integration into host genome. Some viruses, particularly DNA viruses, integrate their genetic material into the host genome. This integration can disrupt normal cell cycle regulation, promote the expression of viral oncogenes, or lead to the inactivation of tumor suppressor genes. For example, integration of HPV DNA into the host genome can disrupt normal epithelial cell growth and contribute to the development of cervical cancer. Oncogene expression. Oncogenic viruses may carry viral oncogenes that can directly promote cell proliferation or survival. These viral oncogenes often mimic the function of host proto-oncogenes, leading to the activation of pathways that promote cell division and inhibit cell death. For instance, the VHONC gene in certain retroviruses can encode proteins that promote unchecked cell division. Inactivation of tumor suppressors. Many oncogenic viruses produce proteins that inactivate key tumor suppressor genes, such as p53 and RB retinoblastoma protein. These tumor suppressors are responsible for controlling the cell cycle and initiating apoptosis in response to DNA damage. Viruses such as HPV and SV40, simian virus 40, produce proteins e.g., E6 and E7 in HPV that bind to and inactivate these tumor suppressor proteins, leading to uncontrolled cell proliferation. Chronic inflammation and immune evasion. Some viruses can cause persistent infections that lead to chronic inflammation. Inflammation can create a microenvironment conducive to tumor development by promoting genomic instability, cell survival, and proliferation. Additionally, viral evasion of the immune system enables the persistent survival of infected cells, allowing time for the accumulation of mutations. Inhibition of apoptosis. Many viruses can inhibit the normal apoptosis pathways in infected cells, allowing damaged cells to survive and proliferate. This inhibition can occur through the expression of viral proteins that block apoptotic pathways, such as the BCL2 family proteins, which are often involved in regulating cell death. Oncogenic viruses and their associated cancers. 1. Human papillomavirus (HPV). HPV is one of the most well-known oncogenic viruses. It is responsible for a variety of cancers, most notably cervical cancer, but also anal, oropharyngeal, and penile cancers. Mechanism. HPV produces E6 and E7 proteins that inactivate p53 and RB tumor suppressors. The E6 protein binds to and promotes the degradation of p53, preventing it from initiating apoptosis in cells with damaged DNA. The E7 protein binds to and inactivates RB, releasing E2F transcription factors, which promote progression through the cell cycle, particularly the G1S checkpoint. Clinical significance. HPV vaccines, such as Gardasil, have been developed to prevent infection with high-risk strains of HPV and reduce the incidence of HPV-related cancers. 2. Hepatitis B virus (HBV) and hepatitis C virus (HCV). Chronic infection with HPV and HCV is strongly associated with the development of hepatocellular carcinoma (HCC), a form of liver cancer. Mechanism. HPV and HCV cause chronic liver inflammation, which can lead to cirrhosis, a predisposing condition for liver cancer. HPV integrates its DNA into the host genome, while HCV, being an RNA virus, can cause persistent infection, leading to chronic inflammation and liver damage. Clinical significance. Antiviral therapies targeting HPV, e.g., nucleoside analogs, and HCV, e.g., direct-acting antivirals, can significantly reduce the risk of liver cancer by clearing the virus and reducing inflammation. 3. Epstein-Barr virus (EBV). EBV is associated with several types of cancers, including Burkitt's lymphoma, nasopharyngeal carcinoma, and Hodgkin's lymphoma. Mechanism. EBV infects B cells and can establish latent infection in these cells. The viral-latent membrane proteins LMP1 and LMP2 mimic cellular signals that promote cell survival and proliferation, inhibiting apoptosis and promoting cell cycle progression. This disruption can contribute to the development of lymphomas and other cancers. Clinical significance. EBV-related cancers are often treated with chemotherapy and radiation, but prevention strategies, such as avoiding EBV infection, are not yet fully developed. 4. Human T-cell lymphotropic virus (HTLV). HTLV is a retrovirus that can cause adult T-cell leukemia, lymphoma, ATL. Mechanism. HTLV carries a tax gene that encodes a protein responsible for activating several cellular pathways that lead to uncontrolled T-cell proliferation. This viral protein stimulates the NF-kappa B pathway, which plays a critical role in inflammation and immune response regulation, thus contributing to the transformation of T-cells into cancerous cells. Clinical significance. HTLV-associated ATL is a rare but aggressive form of leukemia. There is currently no cure for ATL, and treatment typically involves chemotherapy. Cancer prevention and treatment. Vaccination. Vaccines against HPV, e.g., Gardasil and Cervarix, have significantly reduced the incidence of HPV-associated cancers, particularly cervical cancer. Vaccination against HPV, e.g., Injirix B and Recombavax HB, has also been shown to reduce the risk of liver cancer. Antiviral therapy. Antiviral drugs for HPV and HCV can significantly reduce the risk of developing liver cancer by reducing viral load and inflammation. For example, direct-acting antivirals (DAAs) for HCV have revolutionized treatment and significantly improved cure rates. Immune-based therapies. Researchers are exploring immune therapies such as checkpoint inhibitors and therapeutic vaccines to treat virus-induced cancers by stimulating the immune system to target and eliminate infected cells. Previous year questions. Which of the following viruses is most strongly associated with cervical cancer? A. Hepatitis B virus (HBV). B. Epstein-Barr virus (EBV). C. Human papillomavirus (HPV). D. Human T-cell lymphotropic virus (HTLV). Answer. C. Human papillomavirus (HPV). CSIRNET 2016. The E6 and E7 proteins of human papillomavirus (HPV) primarily inactivate which tumor suppressor proteins? A. P53 and RB. B. P16 and P21. C. PRCA1 and P53. D. P53 and P10. Answer. A. P53 and RB. CSIRNET 2019. Which of the following is a common feature of cancers caused by Epstein-Barr virus (EBV)? A. Inactivation of RB protein. B. Chronic inflammation leading to liver cirrhosis. C. Latent infection in B cells. D. Activation of the tax gene. Answer. C. Latent infection in B cells. CSIRNET 2017. Conclusion. Virus-induced cancers are a significant public health issue, and the molecular mechanisms driving virus-induced tumorigenesis are complex. Oncogenic viruses, including HPV, HBV, HCV, EBV, and HTLV, promote cancer through mechanisms such as oncogene activation, tumor suppressor inactivation, chronic inflammation, and immune evasion. Understanding these mechanisms is crucial for the development of targeted prevention and therapeutic strategies to reduce the incidence and mortality of virus-associated cancers. You said, metastasis, ChatGPT said. Metastasis, mechanisms and implications in cancer progression. Metastasis is the process through which cancer cells spread from their original primary site to form secondary tumors in distant organs. This is a hallmark of cancer and the primary cause of cancer-related mortality, as metastatic tumors are often more difficult to treat and manage than primary tumors. The process of metastasis involves several steps, including cell detachment, invasion of surrounding tissues, intravasation into blood vessels or lymphatic vessels, circulation, extravasation into distant tissues, and the formation of secondary tumors in new organs. Understanding the mechanisms of metastasis is crucial for developing effective therapies to prevent or treat metastatic cancer. Steps in the process of metastasis: local invasion. The first step in metastasis is the local invasion of cancer cells into the surrounding tissue. Cancer cells acquire the ability to break free from the primary tumor by degrading the extracellular matrix (ECM), which provides structural support to tissues. This degradation is facilitated by proteolytic enzymes like matrix metalloproteinases (MMPs), plasminogen activators, and cathepsins. The cancer cells alter their adhesion properties by downregulating cell adhesion molecules (CAMs), such as E-cadherin, which normally mediate cell-to-cell adhesion in epithelial tissues. Intravasation. After breaking through the basement membrane, cancer cells enter the bloodstream or lymphatic system through a process called intravasation. The cancer cells must overcome several barriers, including the immune system, endothelial cells, and the sheer stress of the circulating blood. This process is facilitated by the secretion of factors that promote vascular permeability, such as vascular endothelial growth factor (VEGF). Additionally, platelets often surround cancer cells in the bloodstream, helping them evade immune detection. Circulation. Once in the bloodstream or lymphatic system, cancer cells travel through the circulatory system to distant organs. This phase is often referred to as the cancer cell's journey. The majority of circulating cancer cells do not survive the hostile environment of the bloodstream due to immune system attack, lack of nutrients, or mechanical damage. However, a small subset of cancer cells can survive this phase, particularly those that form emboli by clumping with platelets or other cells, which protects them from immune attack. Extravasation. Cancer cells that survive circulation exit the bloodstream and invade new tissues in a process called extravasation. Extravasation occurs when cancer cells interact with the endothelial cells lining the blood vessels of distant organs, allowing them to squeeze through the vessel wall and enter the surrounding tissue. Once in the target tissue, the cancer cells must adapt to the new microenvironment, which often involves additional modifications to their phenotype, enabling them to establish a new tumor. Colonization and formation of secondary tumors. After extravasation, cancer cells can proliferate and form secondary tumors in distant organs. Colonization involves complex interactions between the cancer cells and the microenvironment of the new tissue. This process requires the recruitment of various growth factors and the establishment of a blood supply angiogenesis to support the grown tumor. The seed and soil hypothesis suggests that certain cancers have a predilection for metastasizing to specific organs, e.g., breast cancer often metastasizes to the bone, and lung cancer often metastasizes to the brain due to the compatibility of the seed cancer cell with the soil organ microenvironment. Mechanisms and key factors involved in metastasis: genetic and epigenetic changes. Cancer cells undergoing metastasis often exhibit genetic mutations and epigenetic changes that enhance their invasive and migratory abilities. Mutations in genes that regulate the cell cycle, e.g., p53, RB, cyclins, and signal transduction pathways, e.g., ROS, PI3K, AKT, are common in metastatic cancer cells. Additionally, changes in the epigenome, such as altered DNA methylation patterns and histone modifications, can influence the expression of genes that regulate metastasis. Epithelial to mesenchymal transition, EMT. EMT is a key process in metastasis, wherein epithelial cancer. Mechanisms and key factors involved in metastasis: genetic and epigenetic changes. Cancer cells undergoing metastasis often exhibit genetic mutations and epigenetic changes that enhance their invasive and migratory abilities. Mutations in genes that regulate the cell cycle, e.g., p53, RB, cyclins, and signal transduction pathways, e.g., ROS, PI3K, AKT, are common in metastatic cancer cells. Additionally, changes in the epigenome, such as altered DNA methylation patterns and histone modifications, can influence the expression of genes that regulate metastasis. Epithelial to mesenchymal transition, EMT. EMT is a key process in metastasis, wherein epithelial cancer cells lose their characteristic polarity and adhesion properties and acquire a more mesenchymal-like phenotype, which is associated with increased migratory and invasive abilities. During EMT, cancer cells downregulate E-cadherin, which is crucial for cell-cell adhesion, and upregulate N-cadherin and vimentin, proteins associated with mesenchymal cells. EMT also promotes the expression of enzymes that degrade the ECM, allowing cancer cells to invade surrounding tissues. Angiogenesis. For metastatic tumors to grow and establish secondary tumors, they require a blood supply. This is achieved through angiogenesis, the process of forming new blood vessels. Cancer cells secrete angiogenic factors such as VEGF, vascular endothelial growth factor, and FGF, fibroblast growth factor, to stimulate the growth of blood vessels, providing the necessary nutrients and oxygen for tumor growth. Angiogenesis is essential for the establishment of secondary tumors and is a key target for cancer therapies. Immune evasion. Cancer cells that metastasize must evade the host immune system, which would normally recognize and destroy abnormal cells. Metastatic cancer cells can employ various strategies to evade immune surveillance, such as downregulating major histocompatibility complex (MHC) molecules, secreting immunosuppressive cytokines, or inducing T regulatory cells to suppress immune responses. Tumor microenvironment. The microenvironment surrounding a tumor plays a crucial role in metastasis. This environment consists of various components such as fibroblasts, immune cells, blood vessels, and extracellular matrix (ECM) proteins. Metastatic cancer cells can manipulate the microenvironment to their advantage by recruiting fibroblasts to produce growth factors or ECM components that promote tumor progression. Metastatic niche. A metastatic niche is a specialized microenvironment that supports the survival and growth of metastatic cells in distant organs. This niche is shaped by interactions between cancer cells and the host tissue, including the recruitment of immune cells, blood vessels, and extracellular matrix components. Tumor cells are thought to prepare the metastatic niche in distant organs before they arrive, making it more conducive to the establishment of secondary tumors. Clinical implications and treatment strategies. Metastasis is responsible for the vast majority of cancer-related deaths, as it often leads to widespread disease that is difficult to treat. Early detection and prevention of metastasis are critical for improving survival rates in cancer patients. The following therapeutic strategies are under investigation to target metastatic cancer. Targeting EMT and invasion. Strategies aimed at inhibiting EMT, which is crucial for invasion and metastasis, are being developed. Targeting signaling pathways involved in EMT, such as the TGF-beta and WNT pathways, holds potential for preventing cancer spread. Inhibiting angiogenesis. Drugs that inhibit angiogenesis, such as bevacizumab, an anti-VEGF antibody, are being used in clinical practice to block the growth of blood vessels that supply tumors, thus limiting the growth of metastatic tumors. Immunotherapy. Immune-based therapies, such as checkpoint inhibitors, e.g., nivolumab and pembrolizumab, are being used to activate the immune system against metastatic cancer cells. These therapies aim to reverse the immune suppression that allows cancer cells to escape immune surveillance. Targeting the tumor microenvironment. Therapies that disrupt the tumor microenvironment, such as those targeting fibroblasts, immune cells, or the extracellular matrix, are also being investigated. By modifying the tumor stroma, these treatments aim to hinder the ability of cancer cells to metastasize. Previous year questions. Which of the following is the most critical step in the process of metastasis? A. Intravasation. B. Angiogenesis. C. Local invasion. D. Extravasation. Answer. C. Local invasion. CSIRNET 2017. Epithelial to mesenchymal transition (EMT) is essential for A. Cancer cell proliferation. B. Cancer cell survival. C. Metastasis. D. Angiogenesis. Answer. C. Metastasis. CSIRNET 2018. Which of the following is a major factor promoting angiogenesis in metastatic tumors? A. Vascular endothelial growth factor (VEGF). B. Epidermal growth factor (EGF). C. Platelet-derived growth factor (PDGF). D. Fibroblast growth factor (FGF). Answer. A. Vascular endothelial growth factor (VEGF). CSIRNET 2019. Conclusion. Metastasis is a complex, multi-step process that is central to cancer progression and a major cause of cancer-related mortality. Understanding the molecular mechanisms involved in metastasis, such as local invasion, EMT, angiogenesis, and immune evasion, is crucial for developing targeted therapies to prevent or treat metastatic cancer. Research is ongoing to identify novel therapeutic strategies that can block specific steps in the metastatic process, ultimately improving the prognosis for cancer patients. You said. Interaction of cancer cells with normal cells. ChatGPT said. Interaction of cancer cells with normal cells. Implications for tumor progression. The interaction between cancer cells and normal cells is a critical aspect of tumor progression. While cancer cells often exhibit characteristics that distinguish them from normal cells, they frequently interact with their surrounding normal cells, including stromal cells, immune cells, endothelial cells, and fibroblasts, which contribute to tumor growth, metastasis, and resistance to therapy. The tumor microenvironment (TME) is shaped by these interactions, and these normal cells play an active role in facilitating or hindering cancer progression. Tumor microenvironment (TME). The tumor microenvironment consists of not only cancer cells but also various non-malignant cells, extracellular matrix (ECM) components, blood vessels, and signaling molecules. The TME influences cancer cell behavior, such as proliferation, survival, migration, and resistance to therapies. The interactions between cancer cells and normal cells in the TME are critical for several stages of cancer progression, including angiogenesis, immune evasion, and metastasis. Fibroblasts. Cancer-associated fibroblasts (CAFs) are the most abundant stromal cells in the TME. These fibroblasts undergo activation and acquire characteristics that promote cancer progression. CAFs secrete ECM components like collagen, fibronectin, and proteoglycans, as well as growth factors such as vascular endothelial growth factor (VEGF), fibroblast growth factor (FGF), and transforming growth factor beta (TGF-beta). These factors stimulate cancer cell growth, angiogenesis, and immune suppression. Additionally, CAFs can induce epithelial to mesenchymal transition (EMT) in cancer cells, which enhances their invasive and migratory properties. Immune cells. The immune cells within the TME can have dual roles. They can either suppress or promote cancer progression depending on the type of immune cell involved and the cytokines they secrete. Tumor-associated macrophages (TAMs) are often polarized into an M2 phenotype, which supports tumor growth and metastasis by secreting immunosuppressive cytokines like IL-10 and TGF-beta. TAMs also promote angiogenesis and tissue remodeling, which aid in tumor progression. Regulatory T cells, TREGs are another immunosuppressive cell type that accumulates in the TME. They suppress the immune system's ability to attack cancer cells by secreting IL-10 and TGF-beta, which inhibit the function of cytotoxic T cells. Dendritic cells (DCs) can present tumor antigens to T cells, but in the TME, they are often impaired and unable to initiate a proper immune response against cancer cells due to the immunosuppressive environment. Endothelial cells and angiogenesis. Endothelial cells line the blood vessels, and their interaction with cancer cells is essential for tumor growth and metastasis. As tumors grow, they induce the formation of new blood vessels through a process called angiogenesis. Vascular endothelial growth factor (VEGF) and fibroblast growth factor (FGF) secreted by cancer cells, CAFs, and immune cells stimulate endothelial cell proliferation and migration, leading to the formation of new blood vessels that supply the tumor with oxygen and nutrients. However, the newly formed blood vessels are often leaky and abnormal, contributing to tumor hypoxia and further promoting cancer progression. Extracellular matrix (ECM) components. The ECM is not merely a passive scaffold; it plays a dynamic role in regulating cancer cell behavior. The ECM consists of proteins such as collagen, fibronectin, and laminin, which provide structural support to tissues and organs. Cancer cells interact with the ECM via integrins and other adhesion molecules, which not only anchor them but also provide signals that regulate cell proliferation, survival, and migration. The ECM is often remodeled in the TME by proteases like matrix metalloproteinases (MMPs), which degrade ECM components, allowing cancer cells to invade surrounding tissues and metastasize to distant organs. Adipocytes. Adipocytes, or fat cells, are present in many tumors, particularly those in tissues like the breast and prostate. These cells interact with cancer cells by secreting adipokines such as leptin and adiponectin, which influence tumor growth and progression. Leptin has been shown to promote cancer cell proliferation, migration, and survival, while adiponectin may have anti-cancer properties. Additionally, adipocytes can contribute to the formation of a lipid-rich microenvironment, which can provide energy to cancer cells and facilitate tumor growth. Mechanisms of interaction between cancer cells and normal cell cytokine and growth factor signaling. Cancer cells release a variety of cytokines and growth factors that alter the behavior of surrounding normal cells. For example, cancer cells secrete TGF-beta, which promotes tumor progression by inducing immune tolerance and remodeling the ECM. Interleukins ILs like IL-6 and IL-8 are produced by both cancer and stromal cells and contribute to inflammation and immune suppression in the TME. Growth factors like EGF, epidermal growth factor, and FGF, fibroblast growth factor, can activate signaling pathways like PI3K, AKT, and MAPK, which promote cell proliferation and survival. These factors act in both an autocrine manner on the cancer cells themselves and a paracrine manner on neighboring normal cells, such as CAFs and endothelial cells. Altered adhesion properties. Cancer cells exhibit altered expression of adhesion molecules that mediate interactions with normal cells in the ECM. For instance, the loss of E-cadherin expression in epithelial cancer cells facilitates the detachment of these cells from the primary tumor and their invasion into surrounding tissues. This change is often accompanied by the upregulation of N-cadherin and vimentin, which are characteristic of mesenchymal cells, enabling the cancer cells to migrate and invade. Immune modulation. Cancer cells can modulate the function of normal immune cells to evade immune surveillance. For example, cancer cells can induce the recruitment of TREGs and myeloid-derived suppressor cells, MDSCs, both of which suppress the activation of cytotoxic T cells. This immune evasion is further facilitated by the secretion of TGF-beta, IL-10, and VEGF by both tumor cells and stromal cells. Exosome release and cell-to-cell communication. Cancer cells can release small vesicles known as exosomes, which carry proteins, lipids, and RNA molecules that can influence the behavior of surrounding normal cells. These exosomes are involved in communication between cancer cells and normal cells in the TME and can modulate immune responses, promote angiogenesis, and enhance the invasive properties of cancer cells. Impact of cancer cell interactions with normal cells tumor progression. Interactions between cancer cells and normal stromal cells, such as fibroblasts, macrophages, and endothelial cells, promote tumor growth and invasion. For instance, the secretion of growth factors by CAFs and immune cells can enhance cancer cell proliferation and survival, while the remodeling of the ECM facilitates cancer cell invasion into neighboring tissues. Resistance to therapy. Normal cells within the TME contribute to the development of therapy resistance in cancer. For example, the presence of TAMs and TREGs can suppress the effectiveness of chemotherapy and immunotherapy by creating an immunosuppressive microenvironment. Additionally, endothelial cells in the TME can alter the delivery and efficacy of chemotherapy drugs by forming abnormal blood vessels that hinder drug penetration. Metastasis. The interaction of cancer cells with normal cells is essential for metastasis. Stromal cells such as fibroblasts, immune cells, and endothelial cells play a pivotal role in preparing the pre-metastatic niche in distant organs, making them more susceptible to colonization by cancer cells. In addition, the remodeling of the ECM by cancer-associated fibroblasts CAFs creates pathways that facilitate cancer cell migration and invasion. Previous year questions. Which of the following plays a role in the interaction between cancer cells and stromal cells in the tumor microenvironment? A. Cytokines. B. Growth factors. C. Extracellular matrix remodeling. D. All of the above. Answer. D. All of the above. CSIRNET 2019. Answer. D. All of the above. CSIRNET 2019. The recruitment of regulatory T cells (TREGs) into the tumor microenvironment is primarily responsible for A. Enhanced cancer cell proliferation. B. Immune suppression. C. Angiogenesis. D. Invasion and metastasis. Answer. B. Immune suppression. CSIRNET 2017. Cancer-associated fibroblasts CAFs contribute to tumor progression by A. Producing growth factors. B. Inducing epithelial to mesenchymal transition (EMT). C. Remodeling the extracellular matrix. D. All of the above. Answer. D. All of the above. CSIRNET 2018. Conclusion. The interaction between cancer cells and normal cells is fundamental to the process of tumor progression, metastasis, and resistance to treatment. Cancer cells manipulate their environment by engaging with various normal cell types, including fibroblasts, immune cells, endothelial cells, and adipocytes, to promote tumor growth, angiogenesis, immune evasion, and tissue remodeling. Understanding these interactions is crucial for developing new therapeutic strategies that target the tumor microenvironment and prevent cancer metastasis. You said, apoptosis, ChatGPT said. Apoptosis, mechanisms and regulation. Apoptosis, also known as programmed cell death, is a highly regulated and controlled process that plays a crucial role in maintaining cellular homeostasis and tissue development. Unlike necrosis, which is associated with uncontrolled cell death due to injury or damage, apoptosis is a programmed and orderly process that leads to the removal of unnecessary, damaged, or potentially harmful cells without causing inflammation. It is essential for processes like development, immune system function, tissue homeostasis, and the elimination of cancerous cells. Key features of apoptosis. Apoptosis is characterized by several morphological changes. Cell shrinkage. The cell's volume decreases as it condenses. Chromatin condensation. The chromatin condenses and becomes fragmented, leading to DNA degradation. Membrane blebbing. The plasma membrane forms blebs, which eventually break off. Formation of apoptotic bodies. The cell fragments into small, membrane-bound vesicles called apoptotic bodies, which are phagocytosed by surrounding cells without triggering an inflammatory response. Caspase activation. Caspases, a family of cysteine proteases, are activated and cleave various cellular substrates to orchestrate the dismantling of the cell. Mechanisms of apoptosis. Apoptosis can be initiated through two main pathways, the intrinsic pathway and the extrinsic pathway. Both pathways converge on the activation of executioner caspases, primarily CASPASE3, which are responsible for executing the final steps of apoptosis. 1. Intrinsic pathway, mitochondrial pathway. The intrinsic pathway is triggered by intracellular signals such as DNA damage, oxidative stress, or developmental cues. It is regulated by proteins in the mitochondria. BCL2 family proteins. The BCL2 family plays a central role in regulating mitochondrial outer membrane permeabilization, MOMP, a key event in the intrinsic pathway. These proteins are divided into two categories. Pro-apoptotic proteins includes BAX, BAC, and BAD, which promote apoptosis by causing MOMP. Anti-apoptotic proteins includes BCL2, BCLXL, and MCL1, which inhibit MOMP and prevent apoptosis. When pro-apoptotic signals overwhelm anti-apoptotic signals, BAX and BAC proteins are activated, leading to MOMP and the release of mitochondrial contents into the cytosol. Cytochrome C release. The release of cytochrome C from the mitochondria into the cytoplasm is a pivotal event in the intrinsic pathway. Once in the cytoplasm, cytochrome C binds to APAF1 and PROCASPASE9, forming a complex called the apoptosome, which activates CASPASE9. Caspase activation. CASPASE9 activates CASPASE3, the executioner caspase, leading to the cleavage of various substrates such as DNA fragmentation, membrane blebbing, and cytoskeleton breakdown. 2. Extrinsic pathway, death receptor pathway. The extrinsic pathway is initiated by the binding of death ligands to the respective death receptors on the cell surface. This pathway is activated by external signals, particularly from the immune system or damaged tissue. Death receptors. Death receptors are part of the tumor necrosis factor receptor TNFR superfamily and include receptors like FOS, CD95, TNFR1, and Dr. 4/5. These receptors have intracellular death domains that are crucial for transmitting apoptotic signals when activated. FOS ligand facile and TNF. FOS ligand facile binds to the FOS receptor, and tumor necrosis factor TNF binds to TNFR1, both inducing the formation of a signaling complex known as the death-inducing signaling complex DISC. This complex activates CASPASE8, an initiator caspase. Caspase cascade. CASPASE8 then activates CASPASE3, initiating the execution phase of apoptosis. In some cases, CASPASE8 can cleave BID, a pro-apoptotic BCL2 family protein, which triggers the intrinsic pathway by activating BAX/BAC, leading to mitochondrial release of cytochrome C and further amplification of the apoptotic process. Regulation of apoptosis. Apoptosis is tightly regulated by various proteins, and dysregulation of apoptosis is associated with many diseases, including cancer, autoimmune diseases, and neurodegenerative disorders. The regulation of apoptosis occurs at several levels. Caspase inhibitors. The activity of caspases is regulated by inhibitors of apoptosis proteins IAPs, which bind to caspases and prevent their activation. For instance, XIAP, X-linked IAP, inhibits CASPASE3 and CASPASE9. Survival signals. Signaling pathways like the PI3K-AKT pathway and NF-kappa B pathway promote cell survival by inhibiting apoptotic pathways. These pathways can enhance the expression of anti-apoptotic proteins, e.g., BCL2, or inhibit pro-apoptotic proteins, e.g., BAX. P53 tumor suppressor. The P53 protein is a key regulator of the apoptotic response, especially in response to DNA damage. Upon DNA damage, P53 is activated and induces the expression of pro-apoptotic proteins such as PUMA and NOXA, leading to the intrinsic pathway of apoptosis. Mutations in P53 are common in many cancers, leading to apoptosis evasion and uncontrolled cell proliferation. Death ligand signaling. Death receptors like FOS and TNFR1 are also regulated by their ligands. For example, FASL and TNF are tightly controlled to prevent inappropriate apoptosis of healthy cells. Autophagy. Autophagy can act as a protective mechanism against apoptosis under certain conditions. However, excessive or dysregulated autophagy can promote cell death, and the balance between apoptosis and autophagy is critical for cellular homeostasis. Apoptosis in cancer. Cancer cells often develop mechanisms to evade apoptosis, allowing them to survive in unfavorable conditions, proliferate uncontrollably, and resist therapy. Several strategies by which cancer cells evade apoptosis include overexpression of anti-apoptotic proteins. Overexpression of anti-apoptotic BCL2 family proteins, such as BCL2 and BCLXL, allows cancer cells to resist apoptosis despite the presence of pro-apoptotic signals. Mutations in P53. P53 mutations, which are found in over 50% of cancers, led to the loss of a critical checkpoint in apoptosis, enabling the survival of genetically unstable cells. Inactivation of death receptors. Some tumors downregulate the expression of death receptors, e.g., FOS preventing apoptosis initiated by death ligands like FASL. Increased expression of IAPs. Elevated levels of IAPs, e.g., XIAP, can inhibit caspase activation, allowing cancer cells to evade the apoptotic process. Resistance to chemotherapy-induced apoptosis. Many chemotherapy drugs induce apoptosis through DNA damage. Cancer cells can develop resistance to these drugs by activating survival signaling pathways or by upregulating anti-apoptotic proteins, making them less susceptible to drug-induced cell death. Previous year questions. Which of the following proteins are involved in the intrinsic pathway of apoptosis? A. Cytochrome C. B. FOS ligand. C. CASPASE8. D. Both and C. Answer. A. Cytochrome C. CSIRNET 2017. Which of the following is a characteristic feature of apoptosis? A. Cell swelling. B. Plasma membrane rupture. C. Formation of apoptotic bodies. D. Inflammation. Answer. C. Formation of apoptotic bodies. CSIRNET 2016. The activation of CASPASE9 is a key event in which apoptotic pathway. A. Extrinsic pathway. B. Intrinsic pathway. C. Both and B. D. Necrotic pathway. Answer. B. Intrinsic pathway. CSIRNET 2019. Conclusion. Apoptosis is a vital process that ensures the elimination of damaged, unwanted, or harmful cells without causing inflammation. The two primary apoptotic pathways, intrinsic and extrinsic, are tightly regulated and involve complex molecular signaling. The regulation of apoptosis plays a crucial role in maintaining tissue homeostasis and preventing diseases such as cancer. Cancer cells often evade apoptosis through various mechanisms, making apoptosis resistance a key hallmark of cancer progression. Understanding the molecular pathways and regulation of apoptosis is essential for developing targeted therapies aimed at reactivating cell death in cancer cells. Understanding the molecular pathways and regulation of apoptosis is essential for developing targeted therapies aimed at reactivating cell death in cancer cells. You said. Therapeutic interventions of uncontrolled cell growth. ChatGPT said. Therapeutic interventions of uncontrolled cell growth cancer treatment. Uncontrolled cell growth, often characterized by the unchecked proliferation of abnormal cells, is a hallmark of cancer. This uncontrolled growth can occur due to genetic mutations, dysregulation of cell cycle checkpoints, evasion of apoptosis, and other alterations in cellular signaling pathways. Cancer therapies aim to target these mechanisms to slow or stop the growth of cancer cells, induce their death, and prevent metastasis. Various therapeutic strategies have been developed, ranging from traditional treatments like surgery, radiation, and chemotherapy to more advanced molecular and immunotherapy-based approaches. 1. Chemotherapy. Chemotherapy involves the use of cytotoxic drugs that target rapidly dividing cells, aiming to kill cancer cells or inhibit their proliferation. These drugs work by interfering with cell division, DNA replication, or inducing DNA damage, which ultimately leads to cell death. Chemotherapy is most effective on cancers with high proliferate rates, such as leukemia, lymphoma, and certain solid tumors. Mechanism. Chemotherapy drugs, such as alkylating agents, e.g., cyclophosphamide antimetabolites, e.g., methotrexate, 5-fluorouracil, and topoisomerase inhibitors, e.g., doxorubicin, damage the DNA or inhibit enzymes involved in DNA replication, thereby inducing cell death. Challenges. Chemotherapy is not always specific to cancer cells and can also affect normal, healthy dividing cells, leading to side effects like hair loss, nausea, and immune suppression. 2. Radiation therapy, radiotherapy. Radiation therapy uses high-energy radiation to damage the DNA within cancer cells, leading to cell death. Like chemotherapy, radiotherapy targets rapidly dividing cells, making it effective for treating localized tumors. Mechanism. Ionizing radiation, e.g., X-rays, gamma rays induces DNA breaks, either directly or indirectly through the generation of reactive oxygen species ROS. If the DNA damage is severe enough, the cancer cell will undergo apoptosis or cell cycle arrest. Challenges. While radiotherapy can target tumor cells effectively, surrounding healthy tissues may also be affected, leading to side effects such as skin burns, fatigue, and organ toxicity. 3. Targeted therapy. Targeted therapy involves drugs or other substances that specifically target the molecular abnormalities driving cancer cell growth. These therapies are designed to interfere with specific molecules or pathways that are altered in cancer cells, offering a more precise and less toxic approach than traditional chemotherapy. Mechanism. Targeted therapies include monoclonal antibodies, small molecule inhibitors, and other agents that block specific signaling pathways, such as the EGFR epidermal growth factor receptor pathway, VEGF vascular endothelial growth factor pathway, or the BCR-ABL fusion gene in chronic myelogenous leukemia. Examples. Imatinib targets the BCR-ABL fusion protein in chronic myelogenous leukemia, CML. Trastuzumab Herceptin targets HER2 receptors in HER2-positive breast cancer. Bevacizumab Avastin inhibits VEGF to reduce angiogenesis in various cancers. Challenges. Tumor cells may develop resistance to targeted therapies through mutations in the target molecules or activation of alternative signaling pathways. 4. Immunotherapy. Immunotherapy harnesses the body's immune system to recognize and destroy cancer cells. This type of treatment aims to enhance the immune response to cancer or provide external immune components that can specifically target cancer cells. Mechanism. Immunotherapies include monoclonal antibodies, immune checkpoint inhibitors, cancer vaccines, and adoptive cell therapies. Monoclonal antibodies. These antibodies are designed to bind to specific antigens on cancer cells, marking them for destruction by the immune system. Examples include rituximab targeting CD20 on B cells in lymphoma and trastuzumab targeting HER2 in breast cancer. Immune checkpoint inhibitors. These drugs block immune checkpoint proteins such as PD-1, programmed cell death protein 1, or CTLA-4, which are used by tumors to evade immune detection. Pembrolizumab, Keytruda, and nivolumab Opdivo are PD-1 inhibitors that enhance the immune response against cancers like melanoma, lung cancer, and head and neck cancers. CAR T-cell therapy. Chimeric antigen receptor T-cell therapy involves genetically modifying a patient's T-cells to express receptors that target specific cancer antigens. Kymriah and Yescarta are approved CAR T therapies for leukemia and lymphoma. Challenges. Immunotherapy can lead to immune-related side effects, such as autoimmunity, and not all patients respond to treatment. Additionally, tumors may evolve to suppress immune responses. 5. Hormone therapy. Hormone therapy is used to treat cancers that are influenced by hormones, particularly breast cancer and prostate cancer. Many cancers, such as breast cancer, estrogen receptor positive, and prostate cancer, androgen receptor positive, require hormones to grow. Hormone therapy works by either blocking the hormone receptors or by lowering hormone levels in the body. Mechanism. Tamoxifen, an anti-estrogen drug used in the treatment of estrogen receptor positive breast cancer. It blocks the binding of estrogen to its receptor on cancer cells, preventing their growth. Aromatase inhibitors. These drugs reduce estrogen production in postmenopausal women by inhibiting the enzyme aromatase, e.g., letrozole and anastrozole. Anti-androgens. Drugs such as flutamide and bicalutamide block androgen receptors in prostate cancer, preventing the activation of growth pathways. Challenges. Hormone therapy is not effective in all patients, and resistance can develop over time. 6. Gene therapy. Gene therapy is an emerging field that aims to treat cancer by modifying the genetic material within cancer cells. The goal is to either repair or replace defective genes, introduce tumor suppressor genes, or make the cancer cells more susceptible to other treatments. Mechanism. Gene therapy may involve inserting genes that encode pro-apoptotic proteins or tumor suppressors such as p53 or p164 into the cancer cells to restore normal growth control. Another strategy is gene silencing using RNA interference (RNAi) to knock down the expression of oncogenes or other tumor-promoting factors. Challenges. The delivery of genes to the right cells and tissues and ensuring stable expression remains a major challenge. Additionally, immune reactions can hinder the effectiveness of gene therapy. 7. Surgery. Surgical resection is one of the oldest and most effective treatments for localized cancers. The goal is to physically remove the tumor from the body before it can spread to other parts. Surgery is particularly useful for tumors that have not yet metastasized. Mechanism. Surgical removal of tumors reduces the tumor burden and can provide an opportunity for other treatments such as chemotherapy or radiation therapy to be more effective. Challenges. Surgery is not always possible, especially in cases where the tumor is inoperable due to its location, size, or involvement with critical structures. 8. Angiogenesis inhibitors. Angiogenesis, the process of new blood vessel formation, is essential for the growth and spread of tumors. Tumors secrete angiogenic factors like VEGF, which stimulate blood vessel growth to supply the tumor with oxygen and nutrients. Mechanism. Angiogenesis inhibitors, such as bevacizumab Avastin, target the VEGF pathway to prevent the formation of new blood vessels, thereby starving the tumor of nutrients and oxygen and inhibiting its growth. Challenges. Resistance to angiogenesis inhibitors can develop, and they may lead to side effects like hypertension and bleeding. 9. Cryotherapy and hyperthermia. Cryotherapy uses extreme cold to freeze and kill cancer cells, while hyperthermia involves heating tissues to temperatures that can damage or destroy cancer cells. Mechanism. Cryotherapy is often used for small tumors or precancerous lesions, while hyperthermia can be combined with other treatments like radiation to enhance their effectiveness. Challenges. Both therapies are limited to certain types of tumors, and their success depends on the tumor's location and size. Conclusion. Therapeutic interventions for uncontrolled cell growth, primarily aimed at treating cancer, involve a combination of chemotherapy, radiotherapy, targeted 9. 9. Cryotherapy and hyperthermia. Cryotherapy uses extreme cold to freeze and kill cancer cells, while hyperthermia involves heating tissues to temperatures that can damage or destroy cancer cells. Mechanism. Cryotherapy is often used for small tumors or precancerous lesions, while hyperthermia can be combined with other treatments like radiation to enhance their effectiveness. Challenges. Both therapies are limited to certain types of tumors, and their success depends on the tumor's location and size. Conclusion. Therapeutic interventions for uncontrolled cell growth, primarily aimed at treating cancer, involve a combination of chemotherapy, radiotherapy, targeted therapy, immunotherapy, and other specialized approaches. The choice of therapy depends on the type and stage of cancer, as well as the patient's overall health. While these treatments have significantly improved cancer survival rates, challenges such as treatment resistance, side effects, and the need for personalized medicine remain prominent. Ongoing research into molecular pathways and immune responses continues to drive the development of more effective and less toxic cancer therapies. You said. Innate and adaptive immune system cells and molecules involved in innate and adaptive immunity. ChatGPT said. Innate and adaptive immune system, cells and molecules involved in innate and adaptive immunity. The immune system is a complex network of cells and molecules that defend the body against harmful pathogens, such as bacteria, viruses, fungi, and parasites, as well as against abnormal cells, including cancer cells. It is broadly classified into two major components, innate immunity and adaptive immunity. Both systems work together to provide effective immune responses, with innate immunity acting as the first line of defense and adaptive immunity providing a more specific and long-lasting defense. 1. Innate immune system. The innate immune system is the body's initial, non-specific response to infections and injuries. It is present from birth and provides an immediate defense against pathogens. The key features of innate immunity include its rapid response, lack of memory, and broad specificity. Key cells of the innate immune system. Neutrophils. Neutrophils are the most abundant type of white blood cells in the bloodstream and play a crucial role in the initial defense against bacterial infections. They can phagocytize, engulf, and digest pathogens and release antimicrobial substances. Macrophages. These large phagocytic cells are found in tissues and are derived from monocytes. Macrophages act as antigen-presenting cells, APCs, and they play a significant role in both the innate and adaptive immune systems. They phagocytize pathogens and release cytokines to recruit other immune cells to the site of infection. Dendritic cells. Dendritic cells are professional antigen-presenting cells that act as a bridge between the innate and adaptive immune systems. They capture pathogens and present their antigens to T cells in lymph nodes, thus activating the adaptive immune response. Natural killer NK cells. NK cells are cytotoxic lymphocytes that target and kill virus-infected cells and tumor cells. They do not require prior sensitization to specific antigens and use a range of receptors to identify and eliminate infected or abnormal cells. Eosinophils. These cells are primarily involved in combating parasitic infections and allergic reactions. Eosinophils release granules containing toxic proteins that can damage parasites or infected cells. Basophils. Basophils are involved in allergic responses and inflammation. They release histamine and other pro-inflammatory molecules that contribute to the recruitment of other immune cells to sites of infection or injury. Key molecules of the innate immune system. Pattern recognition receptors (PRRs). PRRs are present on immune cells like macrophages, dendritic cells, and neutrophils. They recognize pathogen-associated molecular patterns (PAMPs) that are commonly found on pathogens but not on host cells. Examples of PRRs include toll-like receptors (TLRs) and nod-like receptors (NLRs). Cytokines. Cytokines are signaling molecules that facilitate communication between immune cells. Key cells of the innate immune system. Neutrophils. Neutrophils are the most abundant type of white blood cells in the bloodstream and play a crucial role in the initial defense against bacterial infections. They can phagocytize, engulf, and digest pathogens and release antimicrobial substances. Macrophages. These large phagocytic cells are found in tissues and are derived from monocytes. Macrophages act as antigen-presenting cells, APCs, and they play a significant role in both the innate and adaptive immune systems. They phagocytize pathogens and release cytokines to recruit other immune cells to the site of infection. Dendritic cells. Dendritic cells are professional antigen-presenting cells that act as a bridge between the innate and adaptive immune systems. They capture pathogens and present their antigens to T cells in lymph nodes, thus activating the adaptive immune response. Natural killer NK cells. NK cells are cytotoxic lymphocytes that target and kill virus-infected cells and tumor cells. They do not require prior sensitization to specific antigens and use a range of receptors to identify and eliminate infected or abnormal cells. Eosinophils. These cells are primarily involved in combating parasitic infections and allergic reactions. Eosinophils release granules containing toxic proteins that can damage parasites or infected cells. Basophils. Basophils are involved in allergic responses and inflammation. They release histamine and other pro-inflammatory molecules that contribute to the recruitment of other immune cells to sites of infection or injury. Key molecules of the innate immune system. Pattern recognition receptors (PRRs). PRRs are present on immune cells like macrophages, dendritic cells, and neutrophils. They recognize pathogen-associated molecular patterns (PAMPs) that are commonly found on pathogens but not on host cells. Examples of PRRs include toll-like receptors (TLRs) and nod-like receptors (NLRs). Cytokines. Cytokines are signaling molecules that facilitate communication between immune cells. They can promote inflammation, recruit immune cells to the site of infection, and stimulate the production of other immune molecules. Key cytokines include interleukins, ILs, tumor necrosis factor (TNF), and interferons (IFNs). Complement system. The complement system consists of a group of proteins that work together to enhance the ability of antibodies and phagocytic cells to clear pathogens. It can also directly lyse pathogens. The complement cascade is activated by antibodies, lectins, or directly by microbial surfaces. Acute phase proteins. These include proteins like C-reactive protein (CRP), mannose binding lectin (MBL), and fibrinogen, which are produced by the liver during acute inflammation and contribute to the immune response by promoting opsonization and pathogen recognition. 2. Adaptive immune system. SARS-CoV-2 virus that binds to the ACE2 receptor is an epitope recognized by neutralizing antibodies. T cell epitopes. These are processed and presented by antigen-presenting cells (APCs) on major histocompatibility complex (MHC) molecules. The epitopes presents definition, types, and role in immune response. An antigen is any substance that is recognized by the immune system as foreign or non-self and triggers an immune response. The immune system identifies antigens as a way to detect and combat pathogens such as bacteria, viruses, fungi, and parasites, as well as abnormal cells, including cancer cells. Antigens are typically molecules or molecular structures present on the surface of these pathogens or on the cells of the body that are infected or transformed. 1. Types of antigens. Antigens can be broadly classified into two major categories based on their origin and structure. Exogenous antigens. These are antigens that originate outside the body, typically from pathogens, allergens, or environmental substances. Examples. Bacterial toxins. Virus particles, e.g., the spike protein of SARS-CoV-2. Pollens or dust mites, allergens. Fungal proteins. Exogenous antigens are typically processed and presented to immune cells by antigen-presenting cells, APCs, like dendritic cells, macrophages, and B cells. Endogenous antigens. These are antigens that originate from inside the body, usually from infected cells or cancerous cells. Examples. Viral proteins produced during infection. Abnormal proteins produced by tumor cells. Self-proteins altered due to mutations, e.g., neoantigens in cancer. Endogenous antigens are processed by the infected cells themselves and presented on the surface through MHC class I molecules to cytotoxic T cells, CD8+ T cells. 2. Structure of antigens. Antigens are typically proteins or polysaccharides, but they can also be lipids or nucleic acids. The critical feature of an antigen is the presence of epitopes, also called antigenic determinants, which are specific regions or molecular structures recognized by the immune system, particularly by antibodies or T cell receptors. Proteins. Most antigens are proteins, and they are highly immunogenic. They are the most common type of antigen in vaccines and pathogens. Example. The spike protein of the SARS. Example. The spike protein of the SARS-CoV-2 virus is a major target for antibodies generated during infection or vaccination. Example. The spike protein of the SARS-CoV-2 virus is a major target for antibodies generated during infection or vaccination. Polysaccharides. Carbohydrate molecules on the surface of pathogens or cells can act as antigens, especially in bacteria. Example. The capsular polysaccharides of bacterial species like Streptococcus pneumoniae are recognized by the immune system. Lipids and nucleic acids. Although less common, some lipids and nucleic acids can function as antigens, especially in autoimmune diseases, where the immune system reacts to the body's own molecules. Example. Cardiolipin, a phospholipid in systemic lupus erythematosus, SLE. 3. Immunogenicity and antigenicity. Immunogenicity refers to the ability of an antigen to provoke an immune response, specifically the production of antibodies and the activation of T cells. Antigenicity refers to the ability of a substance to bind to the products of the immune system, e.g., antibodies or T cell receptors. It does not necessarily induce an immune response, but it can interact with components of the immune system. For an antigen to be immunogenic, it must possess certain characteristics. Size. Larger molecules, e.g., proteins and polysaccharides, are more likely to be immunogenic. Smaller molecules, such as haptens, may not be immunogenic on their own but can become immunogenic when bound to larger carrier proteins. Complexity. Highly complex molecules with diverse structures are more likely to provoke immune responses. Foreignness. The more foreign or different the antigen is from the body's own molecules, the more likely it is to be recognized by the immune system. Accessibility. The epitopes of the antigen must be accessible to immune cells. 4. Epitopes and their role in antigen recognition. An epitope is a specific site on an antigen that is recognized by an antibody or a T cell receptor. Epitopes can be classified into two types. B cell epitopes or antibody epitopes. These are recognized by antibodies. They are usually composed of short sequences of amino acids or sugar residues that are exposed on the surface of the antigen. Example. The part of the spike protein of the SARS-CoV-2 virus that binds to the ACE2 receptor is an epitope recognized by neutralizing antibodies. T cell epitopes. These are processed and presented by antigen-presenting cells, APCs, on major histocompatibility. An antigen is any substance that is recognized by the immune system as foreign or non-self and triggers an immune response. The immune system identifies antigens as a way to detect and combat pathogens such as bacteria, viruses, fungi, and parasites, as well as abnormal cells, including cancer cells. Antigens are typically molecules or molecular structures present on the surface of these pathogens or on the cells of the body that are infected or transformed. 1. Types of antigens. Antigens can be broadly classified into two major categories based on their origin and structure. Exogenous antigens. These are antigens that originate outside the body, typically from pathogens, allergens, or environmental substances. Examples. Bacterial toxins. Virus particles, e.g., the spike protein of SARS-CoV-2. Pollens or dust mites, allergens. Fungal proteins. Exogenous antigens are typically processed and presented to immune cells by antigen-presenting cells, APCs, like dendritic cells, macrophages, and B cells. Endogenous antigens. These are antigens that originate from inside the body, usually from infected cells or cancerous cells. Examples. Viral proteins produced during infection. Abnormal proteins produced by tumor cells. Self-proteins altered due to mutations, e.g., neoantigens in cancer. Endogenous antigens are processed by the infected cells themselves and presented on the surface through MHC class I molecules to cytotoxic T cells, CD8+ T cells. 2. Structure of antigens. Antigens are typically proteins or polysaccharides, but they can also be lipids or nucleic acids. The critical feature of an antigen is the presence of epitopes, also called antigenic determinants, which are specific regions or molecular structures recognized by the immune system, particularly by antibodies or T-cell receptors. Proteins. Most antigens are proteins, and they are highly immunogenic. They are the most common type of antigen in vaccines and pathogens. Example, the spike protein of the SARS-CoV-2 virus is a major target for antibodies generated during infection or vaccination. Polysaccharides. Carbohydrate molecules on the surface of pathogens or cells can act as antigens, especially in bacteria. Example, the capsular polysaccharides of bacterial species like Streptococcus pneumoniae are recognized by the immune system. Lipids and nucleic acids. Although less common, some lipids and nucleic acids can function as antigens, especially in autoimmune diseases, where the immune system reacts to the body's own molecules. Example, cardiolipin, a phospholipid in systemic lupus erythematosus, SLE. 3. Immunogenicity and antigenicity. Immunogenicity refers to the ability of an antigen to provoke an immune response, specifically the production of antibodies and the activation of T cells. Antigenicity refers to the ability of a substance to bind to the products of the immune system, e.g., antibodies or T cell receptors. It does not necessarily induce an immune response, but it can interact with components of the immune system. For an antigen to be immunogenic, it must possess certain characteristics. Size. Larger molecules, e.g., proteins and polysaccharides, are more likely to be immunogenic. Smaller molecules, such as haptens, may not be immunogenic on their own but can become immunogenic when bound to larger carrier proteins. Complexity. Highly complex molecules with diverse structures are more likely to provoke immune responses. Foreignness. The more foreign or different the antigen is from the body's own molecules, the more likely it is to be recognized by the immune system. Accessibility. The epitopes of the antigen must be accessible to immune cells. 4. Epitopes and their role in antigen recognition. An epitope is a specific site on an antigen that is recognized by an antibody or a T cell receptor. Epitopes can be classified into two types. B cell epitopes or antibody epitopes. These are recognized by antibodies. They are usually composed of short sequences of amino acids or sugar residues that are exposed on the surface of the antigen. Example, the part of the spike protein of the SARS-CoV-2 virus that binds to the ACE2 receptor is an epitope recognized by neutralizing antibodies. T cell epitopes. These are processed and presented by antigen-presenting cells, APCs, on major histocompatibility complex MHC molecules. The epitopes presented by MHC class I molecules are recognized by cytotoxic T cells, CD8+, and those presented by MHC class II molecules are recognized by helper T cells, CD4+. Example, a peptide derived from an infected cell's protein might be presented by MHC class I molecules to activate CD8+ T cells, which then kill the infected cells. 5. Antigen presentation and activation of immune cells. Once an antigen is encountered, it is processed and presented to T cells by antigen-presenting cells, APCs, such as dendritic cells, macrophages, and B cells. This process involves the following steps. Phagocytosis or endocytosis. APCs engulf the antigen, breaking it down into smaller peptide fragments. Processing. The peptides are loaded onto MHC molecules for presentation to T cells. MHC class I molecules present endogenous intracellular antigens to CD8+ cytotoxic T cells. MHC class II molecules present exogenous extracellular antigens to CD4+ helper T cells. T cell activation. T cells recognize the antigen MHC complex via their T cell receptors, TCRs, leading to their activation. CD4+ helper T cells release cytokines that enhance the immune response, while CD8+ cytotoxic T cells can directly kill infected cells. 6. Role of antigens in immunological memory. During the primary immune response, the body generates a response to a specific antigen. However, after the pathogen is cleared, some of the activated T cells and B cells differentiate into memory cells. These memory cells can, remember, the antigen and mount a more rapid and robust response upon subsequent exposures. This is the basis for long-lasting immunity, which is central to vaccination. 7. Previous year questions and answers. 1. Which of the following is true regarding the structure of an antigen? A. Antigens are always carbohydrates. B. Antigens always contain epitopes. C. Antigens are only proteins. D. Antigens do not elicit immune response. 7. Previous year questions and answers. 1. Which of the following is true regarding the structure of an antigen? A. Antigens are always carbohydrates. B. Antigens always contain epitopes. C. Antigens are only proteins. D. Antigens do not elicit immune responses. Answer. B. Antigens always contain epitopes. 2021. 2. What type of antigen is presented by MHC class II molecules? A. Endogenous antigens. B. Exogenous antigens. C. Both endogenous and exogenous. D. None of the above. Answer. B. Exogenous antigens. 2020. 3. Which of the following is a primary characteristic of an immunogenic antigen? A. It must be of low molecular weight. B. It must be highly complex and foreign. C. It must be a nucleic acid. D. It does not require T cell activation. Answer. B. It must be highly complex and foreign. 2019. These questions assess understanding of antigen characteristics, their interaction with immune cells, and the role of epitopes in antigen recognition. Understanding these principles is crucial for mastering immune system function and the development of vaccines and immunotherapies. You said. Antigenicity. ChatGPT said. Antigenicity. Definition, determinants, and role in immune response. Antigenicity refers to the ability of a substance to bind specifically to the products of the immune system, such as antibodies or T cell receptors, without necessarily inducing an immune response. It is a critical concept in immunology, as it defines whether a molecule can interact with the immune system, leading to recognition and potential immune activation. While antigenicity allows for the recognition of foreign molecules, it is distinct from immunogenicity, which refers to the ability of an antigen to provoke a full immune response. 1. Characteristics of antigenicity. For a substance to be considered antigenic, it must possess certain characteristics that allow it to bind to antibodies or T cell receptors. These characteristics influence how efficiently the immune system can recognize and respond to the antigen. Molecular size. Larger molecules are generally more antigenic than smaller ones. Larger molecules like proteins and polysaccharides have more surface areas for interaction with the immune system. Molecules with a molecular weight of at least 5,000 daltons tend to be antigenic. Example, proteins, due to their complexity and large size, are the most antigenic molecules, while small molecules like haptens may not be antigenic unless conjugated to larger carriers. Molecular complexity. Antigens with complex, diverse structures are more likely to be antigenic. The diversity in amino acid sequences in proteins or sugar residues in polysaccharides enhances the ability to generate multiple epitopes, increasing the likelihood of immune recognition. Example, the spike protein of the SARS-CoV-2 virus is highly antigenic due to its large size and complex structure, allowing it to present multiple epitopes for immune recognition. Foreignness. The more foreign or different a molecule is from the body's self-components, the more likely it is to be recognized as an antigen. Molecules that closely resemble self-proteins may not be efficiently detected, which is why the immune system can sometimes fail to recognize and attack cancer cells or pathogens. Molecular complexity. Antigens with complex, diverse structures are more likely to be antigenic. The diversity in amino acid sequences in proteins or sugar residues in polysaccharides enhances the ability to generate multiple epitopes, increasing the likelihood of immune recognition. Example, the spike protein of the SARS-CoV-2 virus is highly antigenic due to its large size and complex structure, allowing it to present multiple epitopes for immune recognition. Foreignness. The more foreign or different a molecule is from the body's self-components, the more likely it is to be recognized as an antigen. Molecules that closely resemble self-proteins may not be efficiently detected, which is why the immune system can sometimes fail to recognize and attack cancer cells or pathogens. Example, bacterial proteins are recognized as foreign, whereas human proteins are usually tolerated by the immune system, unless they are altered by mutations, e.g., in cancer. Accessibility. Antigens must be structurally accessible to immune cells, particularly antibodies and T cells. Antigens that are embedded deeply within a pathogen's surface or within cells may be less accessible than those exposed on the surface. Example, viral surface proteins, like the HIV envelope glycoproteins, are accessible and can be targeted by antibodies. Stability. Antigens need to be stable enough to maintain their structure and epitopes so that the immune system can effectively recognize them. Unstable molecules may not be recognized or may degrade too quickly for an immune response to be initiated. 2. Determinants of antigenicity. Antigenicity is largely determined by the epitopes, also known as antigenic determinants, which are the specific regions of the antigen that are recognized by the immune system. These epitopes can be recognized by either antibodies in humoral immunity or T cell receptors in cellular immunity. B cell epitopes. These epitopes are recognized by antibodies, and they typically consist of short amino acid sequences or sugar residues exposed on the surface of the antigen. These epitopes are often conformational, i.e., dependent on the overall 3D structure of the antigen. Example, the epitopes on the spike protein of the SARS-CoV-2 virus are recognized by neutralizing antibodies, which prevent the virus from entering human cells. T cell epitopes. These epitopes are usually presented by antigen-presenting cells, APCs, on major histocompatibility complex (MHC) molecules and are recognized by T cell receptors. T cell epitopes can be linear, consisting of a continuous sequence of amino acids, or nonlinear, formed from distant parts of the protein that are brought together through protein folding. Example, viral peptides presented by MHC class I molecules to cytotoxic T cells, CD8 plus T cells, or by MHC class II molecules to helper T cells, CD4 plus T cells. 3. Haptens and their role in antigenicity. A hapten is a small molecule that alone cannot induce an immune response but becomes antigenic when bound to a larger carrier molecule, such as a protein. This conjugation helps the hapten acquire the necessary size and complexity to be recognized by the immune system. Example, the drug penicillin can act as a hapten. When it binds to proteins in the body, it may form a complex that the immune system recognizes as foreign, potentially leading to an allergic reaction. 4. Immunogenicity versus antigenicity. While antigenicity refers to the ability of a molecule to bind to antibodies or T cell receptors, immunogenicity refers to its ability to provoke a full immune response, including activation of T cells, B cells, and the production of antibodies. In other words, all immunogenic substances are antigenic, but not all antigenic substances are immunogenic. Example, an antigen may bind to an antibody demonstrating antigenicity, but if it does not stimulate a sufficient immune response, e.g., failing to activate helper T cells or to stimulate an adequate production of antibodies, it may not be considered immunogenic. 5. Factors affecting antigenicity. Several factors can influence antigenicity, including environmental factors, such as the way an antigen is introduced into the body, e.g., through injection or ingestion. Dose. The amount of antigen presented to the immune system can impact its ability to induce an immune response. Route of entry. The method of antigen introduction, e.g., via the skin, mucous membranes, or bloodstream, can affect how the immune system recognizes and responds to it. 6. Role of antigenicity in immunotherapy and vaccine development. Understanding antigenicity is crucial for the development of vaccines and immunotherapies. Vaccines often introduce non-infectious versions of antigens, e.g., inactivated virus particles or recombinant proteins, to stimulate the immune system without causing disease. By ensuring that these antigens are highly antigenic, vaccines can train the immune system to recognize and respond to pathogens effectively. Example, in the development of mRNA vaccines, such as the Pfizer-BioNTech and Moderna COVID-19 vaccines, the spike protein of the SARS-CoV-2 virus was selected as an antigen due to its ability to stimulate a strong immune response. 7. Previous year questions and answers. 1. Which of the following factors does not affect the antigenicity of a molecule? A. Molecular size. B. Stability. C. Internal structure. D. Molecular weight. Answer. D. Molecular weight 2019. 2. What is the key difference between immunogenicity and antigenicity? Antigenicity refers to binding to antibodies, while immunogenicity refers to the ability to provoke a full immune response. B. Immunogenicity refers to binding to antibodies, while antigenicity refers to the ability to provoke a full immune response. C. Both terms are interchangeable. D. None of the above. Answer. Antigenicity refers to binding to antibodies, while immunogenicity refers to the ability to provoke a full immune response. 2021. 3. Which of the following is true about epitopes? A. Epitopes are recognized by both antibodies and T cell receptors. B. Epitopes are only recognized by T cell receptors. C. Epitopes are only found in viral proteins. D. Epitopes cannot be recognized by antibodies. Answer. Epitopes are recognized by both antibodies and T cell receptors. 2020. By thoroughly understanding antigenicity, researchers and clinicians can better design vaccines, immunotherapies, and diagnostic tools that effectively utilize the body's immune system to combat diseases. You said. Immunogenicity. ChatGPT said. Immunogenicity. Definition, determinants, and role in immune response. Immunogenicity refers to the ability of a substance called an immunogen to provoke an immune response in the body. Unlike antigenicity, which refers to the ability of a substance to bind to specific antibodies or T cell receptors, immunogenicity specifically refers to the ability of a substance to trigger a full, adaptive immune response that involves the activation of B cells, T cells, and the production of antibodies. The key difference is that all immunogens are antigenic, but not all antigenic substances are immunogenic. An immune response involves the recognition of the immunogen by the immune system, followed by a cascade of immune activities, including the activation of lymphocytes, T cells, and B cells, and the production of antibodies. Immunogenicity is an essential feature for any vaccine or immunotherapy to be effective. 1. Key features of immunogenicity: stimulating adaptive immunity. The process of immunogenicity begins when an immunogen enters the body. For the immune system to react, the substance must be recognized as foreign and capable of stimulating specific immune cells, i.e., T cells and B cells. This typically involves both humoral, antibody-mediated, and cellular T cell-mediated immune responses. Humoral immunity involves the production of antibodies by B cells, which can neutralize the pathogen or mark it for destruction by other immune cells. Cellular immunity involves the activation of cytotoxic T cells, CD8+, which directly kill infected or abnormal cells, and helper T cells, CD4+, that assist in the immune response. Recognition by antibodies and T cells. Immunogens are recognized by antibodies on the surface of B cells or T cell receptors on cells. This recognition is facilitated through the antigen-specific epitopes, which are small, unique regions of the immunogen that are recognized by the immune system. Immunogenicity refers to the ability of a substance called an immunogen to provoke an immune response in the body. Unlike antigenicity, which refers to the ability of a substance to bind to specific antibodies or T cell receptors, immunogenicity specifically refers to the ability of a substance to trigger a full, adaptive immune response that involves the activation of B cells, T cells, and the production of antibodies. The key difference is that all immunogens are antigenic, but not all antigenic substances are immunogenic. An immune response involves the recognition of the immunogen by the immune system, followed by a cascade of immune activities, including the activation of lymphocytes, T cells, and B cells, and the production of antibodies. Immunogenicity is an essential feature for any vaccine or immunotherapy to be effective. 1. Key features of immunogenicity: stimulating adaptive immunity. The process of immunogenicity begins when an immunogen enters the body. For the immune system to react, the substance must be recognized as foreign and capable of stimulating specific immune cells, i.e., T cells and B cells. This typically involves both humoral antibody-mediated and cellular T cell-mediated immune responses. Humoral immunity involves the production of antibodies by B cells, which can neutralize the pathogen or mark it for destruction by other immune cells. Cellular immunity involves the activation of cytotoxic T cells, CD8+, which directly kill infected or abnormal cells, and helper T cells, CD4+, that assist in the immune response. Recognition by antibodies and T cells. Immunogens are recognized by antibodies on the surface of B cells or T cell receptors on T cells. This recognition is facilitated through the antigen-specific epitopes, which are small, unique regions of the immunogen that are recognized by the immune system. Antibodies produced by B cells can neutralize pathogens directly, while T cells can destroy infected or cancerous cells. Duration of immune response. Immunogenicity is also determined by how long the immune system can recognize and respond to the immunogen. The more immunogenic a substance is, the longer the immune system can remember and mount a response against it in future encounters. Memory response. One of the most important features of immunogenicity is the ability to produce immune memory. After an initial encounter with the immunogen, the immune system remembers it through the production of memory cells, B cells and T cells. If the same pathogen is encountered again, the immune system can respond more quickly and effectively, often before the pathogen can cause disease. 2. Factors influencing immunogenicity. Several factors determine how strongly a substance will provoke an immune response. Size of the immunogen. Size of the immunogen. Larger molecules are generally more immunogenic than smaller ones because they have more surface area for immune system recognition. Small molecules, such as haptens, are typically not immunogenic on their own and require conjugation to larger proteins to elicit an immune response. Example, proteins, with their large size and complex structure, are typically more immunogenic than small peptides or lipids. Complexity and diversity. The more complex and diverse the structure of an antigen, the more likely it is to be recognized by the immune system. Immunogens with a variety of epitopes are more likely to activate both B cells and T cells. Example, the spike protein of the SARS-CoV-2 virus contains multiple epitopes, allowing it to induce both humoral and cellular immune responses. Foreignness. The degree to which an immunogen is recognized as foreign or different from the body's own molecules is a key determinant of its immunogenicity. Substances that are highly foreign, e.g., pathogens, toxins, or transplanted tissues, are more likely to induce an immune response. The body's immune system generally tolerates self-antigens, but if these self-antigens become altered, e.g., due to mutations in cancer, they may trigger an immune response. Example, bacterial proteins, when introduced into the body, are seen as foreign and elicit a strong immune response. Degradability and processing. For an immunogen to be effectively presented to the immune system, it needs to be processed by antigen-presenting cells, APCs, such as dendritic cells, macrophages, or B cells. The immunogen must be broken down into smaller peptide fragments for protein antigens and presented on MHC, major histocompatibility complex molecules to T cells. The ability of the substance to be processed and presented influences its immunogenicity. Example, the processing of viral proteins by APCs leads to the presentation of viral epitopes on MHC molecules, triggering the activation of T cells. Route of administration. The way an immunogen is introduced into the body also affects its immunogenicity. Vaccines, for example, are often administered intramuscularly or subcutaneously to facilitate the activation of immune cells at lymphoid tissues. The immune response can vary depending on whether the immunogen is introduced via injection, orally, or through mucosal routes. Example, oral vaccines for diseases like polio stimulate immunity through the gut-associated lymphoid tissue, while injectable vaccines target systemic immune responses. 3. Immunogenicity and vaccines. Understanding immunogenicity is critical for vaccine design. Vaccines aim to introduce a safe version of an immunogen, either killed, attenuated, or recombinant, that can stimulate a protective immune response without causing disease. Vaccine development. A vaccine must contain an immunogen that is sufficiently immunogenic to provoke an immune response. The most common approach in vaccine development involves using protein antigens or inactivated viruses to stimulate immune recognition. Newer approaches include using mRNA vaccines that code for immunogenic proteins. Example, the mRNA vaccines developed for COVID-19, Pfizer-BioNTech, Moderna contain the genetic code for the SARS-CoV-2 spike protein, which is highly immunogenic and stimulates both antibody production and T cell responses. Adjuvants. In some cases, vaccines include adjuvants, substances that enhance the immunogenicity of the immunogen. Adjuvants help to increase the immune response by stimulating immune cells at the injection site or by promoting the presentation of the antigen to the immune system. Example, the aluminum salts used in some vaccines act as adjuvants by enhancing the immune response to the antigen. 4. Previous year questions and answers. 1. Which of the following factors does not influence the immunogenicity of a substance? A. Molecular size. B. Foreignness. C. Genetic similarity to the host. D. Location of the antigen. Answer. D. Location of the antigen, 2018. 2. Which of the following is true regarding immunogenicity? A. Immunogenicity refers to the ability to bind with antibodies or T cell receptors. B. Immunogenicity always involves the activation of B cells. C. Immunogenicity refers to the ability of an antigen to provoke an immune response. D. Immunogenicity does not require the involvement of T cells. Answer. C. Immunogenicity refers to the ability of an antigen to provoke an immune response. 2020. 3. Which of the following features is crucial for an antigen to be immunogenic? A. It must be a hapten. B. It must be of small size. C. It must be foreign and large. D. It must be incapable of binding to MHC molecules. Answer. C. It must be foreign and large. 2021. Understanding immunogenicity is essential for designing vaccines, immunotherapies, and other therapeutic interventions. It is a key to harnessing the immune system's ability to fight infections and diseases, including cancer, by promoting specific immune responses that are capable of providing long-lasting immunity or therapeutic effects. You said, B and T cell epitopes. ChatGPT said. B and T cell epitopes, definition, structure, and recognition. Epitopes, also known as antigenic determinants, are specific regions of an antigen that are recognized by the immune system, specifically by B cells or T cells. The interaction between epitopes and immune receptors triggers an immune response. Understanding the distinction between B cell epitopes and T cell epitopes is crucial for understanding how the immune system identifies and responds to pathogens, foreign molecules, or even cancer cells. 1. B cell epitopes. B cell epitopes, also known as linear epitopes or conformational epitopes, are regions of an antigen that bind to B cell receptors, BCRs, which are antibodies present on the surface of B cells. When a BCR recognizes its specific epitope, the B cell is activated and differentiates into a plasma cell, which produces antibodies against the antigen. Nature of B cell epitopes. Linear epitopes. These are short, continuous sequences of amino acids in the primary structure of a protein. They are recognized by B cells directly without requiring the antigen to undergo conformational changes. Conformational epitopes. These are formed by the three-dimensional folding of the antigen structure. The epitopes recognized by B cells in this case are spatially distant in the linear sequence of the protein but are brought together in the folded form of the antigen. Recognition mechanism. The B cell receptor binds to the epitope directly without the need for antigen processing. Antibody. B and T cell epitopes, definition, structure, and recognition. Epitopes, also known as antigenic determinants, are specific regions of an antigen that are recognized by the immune system, specifically by B cells or T cells. The interaction between epitopes and immune receptors triggers an immune response. Understanding the distinction between B cell epitopes and T cell epitopes is crucial for understanding how the immune system identifies and responds to pathogens, foreign molecules, or even cancer cells. 1. B cell epitopes. B cell epitopes, also known as linear epitopes or conformational epitopes, are regions of an antigen that bind to B cell receptors, BCRs, which are antibodies present on the surface of B cells. When a BCR recognizes its specific epitope, the B cell is activated and differentiates into a plasma cell, which produces antibodies against the antigen. Nature of B cell epitopes. Linear epitopes. These are short, continuous sequences of amino acids in the primary structure of a protein. They are recognized by B cells directly without requiring the antigen to undergo conformational changes. Conformational epitopes. These are formed by the three-dimensional folding of the antigen structure. The epitopes recognized by B cells in this case are spatially distant in the linear sequence of the protein but are brought together in the folded form of the antigen. Recognition mechanism. The B cell receptor binds to the epitope directly without the need for antigen processing. Antibodies, which are essentially secreted forms of the BCR, also bind to these epitopes on the pathogen or antigen, marking it for neutralization or phagocytosis. Example: the spike protein of the SARS-CoV-2 virus contains B cell epitopes that trigger the production of antibodies in response to vaccination. 2. T cell epitopes. T cell epitopes are short peptide fragments derived from antigens that are presented on the surface of antigen-presenting cells, APCs, by major histocompatibility complex (MHC) molecules. T cell receptors (TCRs) on T cells recognize these peptide MHC complexes. T cells are classified into CD4 plus T helper cells and CD8 plus cytotoxic T cells, with each recognizing different types of epitopes. Types of T cell epitopes: CD4 plus T cell epitopes, MHC class II epitopes. These epitopes are processed from the antigen, typically from proteins, by antigen-presenting cells such as dendritic cells, macrophages, or B cells. The epitopes are presented on the surface of these cells in association with MHC class II molecules. CD4 plus T cells recognize these MHC class II peptide complexes and become activated, triggering a helper immune response. These cells assist other immune cells, including B cells for antibody production and CD8 plus T cells for cytotoxic activity. CD8 plus T cell epitopes, MHC class I epitopes. These epitopes are also processed from intracellular proteins, such as viral proteins or abnormal proteins from cancer cells. These peptides are presented by MHC class I molecules on the surface of all nucleated cells. CD8 plus T cells, which are cytotoxic, recognize these peptide MHC class I complexes and, upon activation, kill the infected or cancerous cells displaying the foreign or mutated peptides. Recognition mechanism. Antigen processing. In both cases, the antigen must be broken down into smaller peptide fragments by the host cell's machinery. CD4 plus T cells recognize extracellular peptides that have been ingested by APCs. CD8 plus T cells recognize intracellular peptides derived from pathogens or mutated proteins in infected or cancerous cells. MHC presentation. The peptides are then presented on the cell surface by MHC molecules, allowing T cells to recognize them. TCR binding. The T cell receptor TCR on CD4 plus or CD8 plus T cell specifically recognizes the peptide MHC complex, leading to T cell activation and the subsequent immune response. 3. Features of T and B cell epitope size. B cell epitopes are typically short, 5-10. Understanding the distinction between B cell epitopes and T cell epitopes is crucial for understanding how the immune system identifies in response to pathogens, foreign molecules, or even cancer cells. 1. B cell epitopes. B cell epitopes, also known as linear epitopes or conformational epitopes, or regions of an antigen that bind to B cell receptors, BCRs, which are antibodies present on the surface of B cells. When a BCR recognizes its specific epitope, the B cell is activated and differentiates into a plasma cell, which produces antibodies against the antigen. Nature of B cell epitopes. Linear epitopes. These are short, continuous sequences of amino acids in the primary structure of a protein. They are recognized by B cells directly without requiring the antigen to undergo conformational changes. Conformational epitopes. These are formed by the three-dimensional folding of the antigen structure. The epitopes recognized by B cells in this case are spatially distant in the linear sequence of the protein but are brought together in the folded form of the antigen. Recognition mechanism. The B cell receptor binds to the epitope directly without the need for antigen processing. Antibodies, which are essentially secreted forms of the BCR, also bind to these epitopes on the pathogen or antigen, marking it for neutralization or phagocytosis. Example: the spike protein of the SARS-CoV-2 virus contains B cell epitopes that trigger the production of antibodies in response to vaccination. 2. T cell epitopes. T cell epitopes are short peptide fragments derived from antigens that are presented on the surface of antigen-presenting cells, APCs, by major histocompatibility complex (MHC) molecules. T cell receptors (TCRs) on T cells recognize these peptide MHC complexes. T cells are classified into CD4 plus T helper cells and CD8 plus cytotoxic T cells, with each recognizing different types of epitopes. Types of T cell epitopes: CD4 plus T cell epitopes, MHC class II epitopes. These epitopes are processed from the antigen, typically from proteins, by antigen-presenting cells such as dendritic cells, macrophages, or B cells. The epitopes are presented on the surface of these cells in association with MHC class II molecules. CD4 plus T cells recognize these MHC class II peptide complexes and become activated, triggering a helper immune response. These cells assist other immune cells, including B cells for antibody production and CD8 plus T cells for cytotoxic activity. CD8 plus T cell epitopes, MHC class I epitopes. These epitopes are also processed from intracellular proteins, such as viral proteins or abnormal proteins from cancer cells. These peptides are presented by MHC class I molecules on the surface of all nucleated cells. CD8 plus T cells, which are cytotoxic, recognize these peptide MHC class I complexes and, upon activation, kill the infected or cancerous cells displaying the foreign or mutated peptides. Recognition mechanism. Antigen processing. In both cases, the antigen must be broken down into smaller peptide fragments by the host cell's machinery. CD4 plus T cells recognize extracellular peptides that have been ingested by APCs. CD8 plus T cells recognize intracellular peptides derived from pathogens or mutated proteins in infected or cancerous cells. MHC presentation. The peptides are then presented on the cell surface by MHC molecules, allowing T cells to recognize them. TCR binding. The T cell receptor TCR on CD4 plus or CD8 plus T cells specifically recognizes the peptide MHC complex, leading to T cell activation and the subsequent immune response. 3. Features of T and B cell epitope size. B cell epitopes are typically short, 5-10 amino acids for linear epitopes and 10 to 15 amino acids for conformational epitopes. T cell epitopes are even shorter, usually 8 to 10 amino acids for CD8 plus T cells and 13 to 25 amino acids for CD4 plus T cells. Location. B cell epitopes are generally found on the surface of proteins, exposed to the extracellular environment. T cell epitopes are typically inside cells, either in the cytoplasm for intracellular pathogens or internalized by the APCs for extracellular pathogens. Processing requirement. B cell epitopes can be recognized directly by BCRs or antibodies without any processing. T cell epitopes must undergo processing by antigen-presenting cells to generate the peptide fragments that are then displayed on MHC molecules. 4. Immunological significance. Immune activation. The activation of B and T cells by their respective epitopes leads to various immune responses, including the production of antibodies by B cells and the activation of cytotoxic T cells by CD8 plus T cells to kill infected or abnormal cells. Vaccine design. Knowledge of B and T cell epitopes is crucial for designing vaccines. Vaccines often aim to introduce known B-cell epitopes to generate antibody responses or T-cell epitopes to generate cellular immunity to protect against specific pathogens or diseases, such as cancer. 5. Previous year questions and answers. 1. Which of the following is an essential feature of T-cell epitopes? A. They can be directly recognized by antibodies. B. They require processing by antigen-presenting cells. C. They are typically large proteins. D. They are exclusively recognized by CD4 plus T cells. Answer. B. They require processing by antigen-presenting cells. 2019. 2. What is the primary role of B-cell epitopes in the immune response? A. Activation of T cells. B. Binding to antibodies for neutralization. C. Killing infected cells. D. Processing antigens into peptides. Answer. B. Binding to antibodies for neutralization. 2020. 3. Which MHC class is associated with the presentation of antigens to CD8 plus T cells? A. MHC class 1. B. MHC class 2. C. MHC class 3. D. Both MHC class 1 and 2. Answer. A. MHC class 1. 2021. Conclusion. The B-cell epitopes and T-cell epitopes are critical elements in the immune response. B-cell epitopes mainly facilitate the production of antibodies by recognizing foreign antigens directly, while T-cell epitopes require processing and presentation by antigen-presenting cells, leading to cellular immune responses. Understanding the distinction and recognition mechanisms of these epitopes is crucial for designing vaccines, therapies, and understanding immune responses to pathogens and cancers. You said. Structure and function of antibody molecules. ChatGPT said. Structure and function of antibody molecules. Antibodies, also known as immunoglobulins (IG), are glycoproteins produced primarily by plasma cells, differentiated B cells, in response to the presence of antigens. Their primary function is to recognize and neutralize foreign objects such as bacteria, viruses, and toxins. Antibodies are essential components of the adaptive immune system, providing specific immunity by binding to and marking pathogens for destruction or neutralization. 1. Structure of antibody molecules. An antibody is typically composed of 4 polypeptide chains, 2 identical heavy chains H, and 2 identical light chains L. The overall structure is shaped like a Y. A. Polypeptide chains. Heavy chains. These are larger polypeptide chains that contribute to the bulk of the antibody structure. There are 5 major types of heavy chains corresponding to the different classes of immunoglobulins IgA, IgD, IgG, and IgM. The heavy chain consists of one. ChatGPT said. Structure and function of antibody molecules. Antibodies, also known as immunoglobulins (IG), are glycoproteins produced primarily by plasma cells, differentiated B cells, in response to the presence of antigens. Their primary function is to recognize and neutralize foreign objects such as bacteria, viruses, and toxins. Antibodies are essential components of the adaptive immune system, providing specific immunity by binding to and marking pathogens for destruction or neutralization. 1. Structure of antibody molecules. An antibody is typically composed of 4 polypeptide chains, 2 identical heavy chains H, and 2 identical light chains L. The overall structure is shaped like a Y. A. Polypeptide chains. Heavy chains. These are larger polypeptide chains that contribute to the bulk of the antibody structure. There are 5 major types of heavy chains corresponding to the different classes of immunoglobulins IgA, IgD, IgG, and IgM. The heavy chain consists of variable region VH, the part of the heavy chain that forms part of the antigen binding site and varies between antibodies, providing specificity to antigen recognition. Constant region CH, the part of the heavy chain that remains the same within each class of antibody and determines the effector functions of the antibody, e.g., activating complement, binding to receptors on immune cells. Light chains. These smaller chains also consist of a variable region VL and a constant region CL. There are 2 types of light chains, kappa kappa and lambda lambda. Light chains are associated with the heavy chains to form the complete antibody structure. B. Antigen binding sites. Each antibody contains 2 antigen binding sites. These sites are formed by the variable regions of both the light and heavy chains, specifically the VH and VL domains. The region of the antibody responsible for binding the antigen is called the paratope, which is complementary in shape to a part of the antigen known as the epitope. C. Fab and FC regions. Fab fragment antigen binding. The Fab region is composed of the variable regions of both the heavy and light chains. It is responsible for the antigen binding activity of the antibody. The Fab fragment is the portion of the antibody that binds to the antigen's epitope, determining the specificity of the antibody. FC fragment crystallizable. The FC region is made up of the constant regions of the heavy chains. It is responsible for interacting with immune effector cells, such as macrophages, neutrophils, and dendritic cells, and activating the complement system. The FC region is involved in the biological activities of the antibody, including opsonization, antibody-dependent cell-mediated cytotoxicity (ADCC), and complement activation. 2. Classes of antibodies. Immunoglobulins. Antibodies are divided into 5 main classes based on differences in their heavy chains. Each class has distinct functions in the immune response. IgG. The most abundant antibody in blood and extracellular fluid. Provides long-term immunity after infection or vaccination. Functions in neutralization, opsonization, and activating the complement system. IgG1, IgG2, IgG3, and IgG4 are subclasses with slightly different roles. IgA. Predominantly found in mucosal areas, e.g., respiratory and gastrointestinal tracts, and secretions, e.g., saliva, breast milk. Protects mucosal surfaces by neutralizing pathogens before they can invade tissues. Exists as monomers in blood and dimers in secretions. IgM. The first antibody produced during an initial immune response. Found mainly in the blood and lymphatic fluid. Functions in agglutination and complement activation. Exists as a pentamer, which enhances its ability to bind antigens. IgG. Associated with allergic reactions and responses to parasitic infections. Binds to mast cells and basophils, leading to the release of histamine and other mediators during an allergic reaction. IgD. Found in small amounts in the blood and is mainly expressed on the surface of naive B cells. Functions primarily in the activation and regulation of B cells. 3. Functions of antibody molecules. Antibodies perform a variety of functions that contribute to the immune defense against pathogens. The major functions are neutralization. Antibodies neutralize pathogens by binding to key components such as toxins, viruses, or bacterial surface proteins, preventing them from interacting with host cells. For example, neutralizing antibodies can bind to viral surface proteins, preventing the virus from entering host cells. B. Opsonization. Opsonization refers to the process by which antibodies coat pathogens, making them more easily recognized and ingested by phagocytic cells, e.g., macrophages, neutrophils. The Fc region of the antibody binds to Fc receptors on phagocytes, enhancing phagocytosis. C. Complement activation. The Fc region of antibodies, particularly IgM and IgG, can bind to components of the complement system. This triggers a cascade of events that lead to the formation of a membrane attack complex, MAC, which can lyse pathogens or infected host cells. Additionally, complement activation leads to the generation of smaller molecules that act as chemotactic signals, attracting immune cells to the site of infection. D. Antibody-dependent cellular cytotoxicity, ADCC. In ADCC, antibodies bind to infected or abnormal cells, marking them for destruction by immune cells such as natural killer NK cells. The Fc region of the antibody interacts with Fc receptors on NK cells, which then release cytotoxic molecules that kill the target cell. E. Agglutination. Antibodies can cross-link multiple antigens, causing them to clump together, a process known as agglutination. This is particularly important for the clearance of pathogens from the bloodstream, as agglutinated pathogens are more easily cleared by the immune system. F. Passive immunity. Antibodies can be transferred from one individual to another, providing passive immunity. For example, IgG antibodies are transferred from mother to fetus through the placenta, providing protection against pathogens during the early months of life. Similarly, antibodies can be administered therapeutically, as seen in the use of monoclonal antibodies for treating certain diseases. 4. Previous year questions and answers. 1. Which part of the antibody molecule is responsible for binding to antigens? A. Fab region. B. FC region. C. Heavy chain. D. Light chain. Answer. Up Fab region. 2019.